I welcome you all to today's session. So let me introduce myself. I'm Dr.Chrisanne Freeman, Assistant Professor, Department of Biotechnology, Bishop Heber College. So today I've chosen the topic of discussion on restriction enzymes, which is also called the molecular scissors of genetics. So let us first dwell into the discovery of restriction enzymes. So the term restriction enzyme was originated from the studies of the lambda-phage, and it was in 1978 that Weena Aber, Daniel Nathans, Hamilton O. Smith, who won the Nobel Prize for the characterization and discovery of the restriction enzymes while they were studying the enterobacteriophage. So let us just recall the structure of a bacteria and the bacteriophage. So we all know that the bacteriophage, the lytic and the lysogenic cycle. So when the phage enters or infects the bacteria cell, the bacterian cell, it transfers its DNA into the bacteria and it replicates its own DNA in the bacterial cell. So now, as a self-defense, these bacterial cells are producing some enzymes called, specific enzymes called restriction enzymes. So as I said, these restriction enzymes are produced from bacterial cells as a self-defense mechanism. So let us see what are restriction enzymes. The restriction enzyme is actually a protein and it is produced from bacteria, and their major role is to cleave the DNA at specific sites. So when I say cleave, it means to cut the DNA at a specific site. Now what are these specific sites? These specific sites are called as restriction sites. Now these restriction sites are unique sites that are present in the DNA molecule. Now these enzymes will go and sit on those specific restriction sites and they cleave the DNA, or in other words, cut the DNA. Now the role of the restriction enzyme is to protect the live bacteria from these bacteriophages. So they cleave the restriction sites of the bacteriophage DNA and destroy its DNA, thus preventing the replication of the viral DNA into the host, which is the bacteria. So these restriction enzymes are employed as important tools in genetic engineering, and they can be isolated from the bacteria and they are used for various genetic engineering applications in the laboratory. So now these are the restriction sites. Can you see? This is the particular site where the bacteria cleaves the DNA molecule. The bacteria cleaves the DNA molecule. So the restriction enzymes cleave the double-stranded DNA molecules at these specific sites, maybe at the adenine, before the adenine base, here and here as well. Now I said defense mechanism in bacteria. So all of us have our own defense mechanism. Protecting our own immune system protects our body from various infections. A similar phenomenon is the production of restriction enzymes by bacteria. So these bacteria produce these restriction enzymes as a defense mechanism against the invading bacteriophages. So now there's a question that arises. If these restriction enzymes are going to cleave a DNA molecule, won't it cleave the own bacterial DNA? That's another question. Yes. So the bacterial DNA cannot be cleaved by these enzymes, and therefore the bacterial DNA modifies its own DNA. The bacteria modifies its own DNA by adding methyl groups at specific sites, especially at the restriction sites where these enzymes try to cleave the bacteria. So now when these methyl groups are added to the bacterial DNA is modified, and therefore these enzymes do not cleave the bacterial DNA. Rather, they identify the invading virus particle, the DNA of the virus particle, and then they are cleaved and degrade it. So this is the major mechanism. So this is the DNA cleavage, where it is very specific and it cleaves specific DNA sequences, which thereby disrupts the phage's genetic material and hence prevents the infection. Let us see the mechanism. I explained the mechanism. I would like to just represent it as a pictorial representation. So just imagine that this is the DNA molecule, the double-stranded DNA molecule. Yes. Then you have a specific enzymes called methyltransferases. These methyltransferases are responsible for addition of methyl groups in the respective recognition site present in the double-stranded DNA molecule. So you can see here, this is called the addition, a simple addition of a methyl group at a base pair. For example, here the methylation occurs at the adenine site. So this is GATC CTAG. So this is considered as the specific recognition site. So you can see here, this is the methyl group and this yellow represents the restriction enzyme. So the methylation occurs at very specific sites of the DNA where the restriction enzymes try to cleave or cut the DNA. So once the DNA of the bacterial host is modified by the simple DNA methylation with the help of the enzyme DNA methyltransferase, there is a modification of the recognition sequence, and therefore the restriction enzymes are not able to cleave the methylated DNA, and rather they cleave the non-methylated DNA. I hope you understood. So next, let's move to the next slide, which talks about the types of restriction enzymes. Now coming to the type of restriction enzymes. So as I said, restriction enzymes cleave at specific restriction sites, which are nothing but specific restriction sequences. Now based on the type of these restriction sequences, where the restriction enzymes cut the particular DNA, whether the restriction enzymes are going to cut at the point of the recognition sequence, or will it cut a few base pairs downstream away from the restriction sequences. So based on where the restriction enzymes cleave the DNA, they are classified into three types: type 1 restriction enzymes, type 2 and type 3 restriction enzymes. So let us just go through the properties. So first, the protein structure. The type 1 is a multi-subunit complex, followed by the type 2, it is multi-enzyme complex, and type 3 again, it's a multi-subunit complex. Coming to this cleavage site, so where it cuts, right? So type 1, it cuts away from the restriction, 1000 base pair downstream away from the restriction site. So this is the restriction site. The enzyme does not cut or cleave the DNA, rather it moves further down. And the type 2, now this type 2 is of genetic engineering, it's very important, okay? Actually, the type 2 enzymes play a very vital role as molecular scissors, and we use them. We will discuss the applications of this in the next few slides. So this type 2 is very important because it cuts at the point of recognition, at the very site of the specific recognition sequence, and hence make a note of it, it's very important. And the type 3 multi-subunit complex enzyme is again does not cut at the specific site, rather cuts away 24 to 26 base pairs downstream. So what do you need for this? Apart from the restriction enzyme, what else do you need? So the requirements for cleavage is, the type 1 requires 2 recognition sequences in any orientation. The type 2 requires 1 recognition sequence, and again the type 3 is similar to that of the type 1. Cofactors, we will look at the restriction enzyme type 2, magnesium as a cofactor. Now what is the role of magnesium? It helps in two ways. It helps to bind the restriction enzymes at the particular bases, especially at the phosphate groups, and also it helps in stabilizing the protein and DNA complex. So you should remember that the restriction enzyme is nothing but a protein. So when the protein or the restriction enzyme binds to the DNA to cleave it, this becomes a protein DNA complex. So it should be stable. So magnesium ion binds to the DNA and makes this subunit, the protein DNA complex, stable. Right? So what is preferred in genetic engineering is the type 2 restriction enzymes. This is preferred for cloning and genetic engineering, while the type 1 and the type 2 are not preferred for cloning. Yes. Now coming to the nomenclature. So these the restriction enzymes have specific nomenclature. You might wonder, what are these funny names that crop up? Yes. So let's take a common example of a restriction enzyme, EcoR1, right? So the recognition sequence is GAATTC. So let's see how they name this. So the first letter is written in capital letter, right? Let's remember that, where in the case of EcoR1, E represents the genus of the bacteria from where the restriction endonuclease has been isolated. So here in the case of EcoR1, it is Escherichia. So E stands for Escherichia. The second two letters represents the species of the bacteria. So it is written as CO in small letter, followed by the third letter R, which represents the strain of the bacteria, which varies from species to species. And the fourth one, the last number is I, which is none other than, it signifies the order of the identification of the enzyme. So if it is going to be the first one to be identified, the first restriction enzyme in this particular species, then it is to be written as 1. So it's a very simple representation which can be globally followed so that confusion does not arise. Let us now concentrate on the type 2 restriction endonucleases. Coming to the type 2 restriction endonucleases, when it cleaves the DNA or cuts the DNA, it does it in two patterns. The first pattern is called as the sticky ends, and the second pattern that it cuts is the blunt ends. So what are sticky ends? Sticky ends are nothing but when these enzymes cut the DNA specifically at a specific recognition site, they cut them asymmetrically, right? So leaving overhangs, single-stranded overhangs, right? So this is called as cohesive ends or sticky ends. And the other type of cut that the type 2 enzymes would also do in certain molecules, DNA molecules, is that it will cut them equally, leaving no overhangs. So this is called as blunt end restriction endonucleases. Now these sticky ends or cohesive end, right? This type of cut is very important. This is what we prefer in genetic engineering. This is what this overhangs will help us to easily clone our gene of interest. So because these overhangs are complementary to each other. So this serves as a very important tool which helps us to easily clone our gene of interest into another host. So let us see an example of cohesive end, EcoR1. Hope you can see the specific double-stranded DNA molecule, which is represented in purple, the recognition sequence, and the EcoR1 enzyme in green, yes, and the red arrow indicating the specific site at which the EcoR1 cleaves the DNA, and it leaves a sticky end. On the other hand, there's another enzyme SMA1, right? So this cuts the DNA of interest with a blunt end. So we go for sticky ends. As I said, they are very important in tools in genetic engineering. Now coming to the applications. Restriction enzymes are considered as key players in genetics. Why? Because number one, they help in gene cloning. You can cut any DNA molecule of interest and you can insert genes, target genes of your interest and express them in another host. So gene expression analysis is one of the very important applications of restriction enzymes. Then coming to RFLP analysis. In case of, especially in fields of molecular diagnostics, in DNA fingerprinting, in identifying mutations, and also for cutting DNA into fragments. Restriction enzymes play a very important role. Of course, another important role is gene or chromosome mapping. Determining the order of genes, how they are located on a particular DNA for a particular organism is very essential because we need to know the function, where the gene is located, on which chromosome, and only if we have that information, we will be able to engineer another molecule of importance. So thank you. I hope you know about restriction enzymes, and these are my references.